So good afternoon, everyone. I'm Avi Reichenberg. I'm a professor here at the Department of Psychiatry, and I'm the host for today's Psychiatry Virtual Lecture Series. We have one of our own speaker today. Dr. Nelly Alia-Klein is an associate professor of psychiatry and neuroscience. She started her training at the Columbia University, and where she received her PhD. She then trained in neuroimaging and addiction in Nora Volkov groups in Brookhaven National Labs, and then joined Mount Sinai in 2013, and she's now the co-chief of the NARC program, the neuropsychoimaging of addiction and related conditions, for those of you who don't know what NARC stands for. She led some very, very innovative studies, and she's world known for her work on the role of MAOA in aggression and addiction, and also did one of the first, if not the first, study of gene by disease interaction. If this is something you haven't heard much about, you've heard a lot about gene environment interaction. It's a version of the gene environment field, worth reading and learning a little bit about after the talk, because I don't think she's going to speak about that today. She will present PET and MRI studies on important but often overlooked contributors to individual differences in addiction. And while you're listening and taking notes and having questions, please put them in the Q&A box at the bottom of the Zoom screen, and we will address them, as many of them as possible, at the end of the talk. So go ahead, Nelly, it's all yours.
Okay, thank you. It's wonderful to be here. It's a great forum. I have to congratulate it because it really gives us a chance as faculty to present to each other, faculty and trainees to present to each other and see what we're doing, and maybe learn from each other. I'm going to share my screen here. There we go. And start the presentation. Okay, can you see it? Yeah. Okay, so what is drug addiction? Drug addiction is a disorder, a chronic disorder, where there's a host of symptoms that go around in a vicious cycle. It starts with a particular sensitivity to certain drugs and drug cues, especially in the context of stress. It involves craving for the drug or drugs, and also episodes of loss of control, consumption of the drug, episodes of withdrawal. And so the drug is becoming very reinforcing over time. And there is impaired response inhibition, which is essentially impairment in self-control. And it is especially problematic in the context of the hypersalience or the hyperimportance that is attributed to the drug of abuse. And so you have these two processes happening there. And in the last 2 decades, Rita and I have been studying these, and my focus was a lot of individual differences, because it always was daunting to see the heterogeneity of the brain regions that we're seeing in the results, and thinking what might be other contributions to this immense variability that we're seeing in brain and behavior. And so one of the important variable of individual differences is age. Most of the people that we study are adults in their 40s. And so we don't get to study children. We don't get to study new to exposures. We don't get to study older adults. And when I say we don't get to study, I'm talking about us also as a lab, but also the field of addiction research. There are gaps, and there are gaps in age and development, and perhaps especially at older age. Sex and gender. Of course, I'm going to be talking a lot about sex and gender. Sex and gender is actually the first part of my talk. Of course, personality traits that develop well before the onset of addiction, or they develop, the temperaments develop well before the onset of addiction. And genetic variability, and here I'm talking about genetic polymorphisms. So common genetic variability in humans, and other drug use. Very often, other drug use is already chronic in individuals that were studying for cocaine or heroin addiction. And childhood trauma, a source of stress that might very well be before the onset, pre-morbid, before the onset of addiction. So you ask yourself, what are the factors, the different factors that contribute to phenotype expression? And when I think about addiction, I think about addiction in terms of severity. So not just the factors vary, but also addiction varies. And one of the ways in which it varies is in severity, especially if over the years, if you have many, many years of drug use. And so on the y-axis here, you have addiction severity, and here on the x-axis, you can see the lifespan. So it starts with genetic contribution, of course, that is inherited, but also in utero exposures. In many individuals with addiction, there is a history of hyperactivity and emotional problems that are pre-morbid. There's a history of stress, including childhood abuse, childhood trauma, and neglect. Neglect is very important, and it's neglected in the literature. Unfortunately, I will not be talking about neglect. I'll be talking about childhood trauma. And then you have, in adolescence, the use of drugs. Sometimes it starts in childhood. Of course, this figure is just sort of a graphic to give you a broad stroke, yes? But there are a lot of substances that start even before addiction to the primary substance that we're looking at, for example, cocaine or heroin. Smoking is a very, very important factor. It's a factor that inhibits, the chronic smoking inhibits monoamine oxidase A, which are major, and B, which are major regulators of monoamine in the brain and in the whole body. And also it has to do with inhibition of aromatase, which is a very important gonadal hormone that is also active in the brain. And alcohol does very similar things. So you're thinking about these substances and in turn their effects during adolescence. And as the person is developing the full-blown addiction that we are studying. And so you can see adolescence, their puberty, sex hormones, and a lot of other issues. There's a problem in decision-making. It's already starting to be impaired because of the addiction, because of the use of other drugs. High anger, negative emotions, poor inhibitory control. And when we study, these individuals, we study addiction at this age, adulthood. Our samples are about 35 to 45 years old. And so we study them when actually they have within them all these aggregate effects of addiction that contributed in turn to the onset, but also to the severity of addiction. And there's a lot of data backing all of these different factors. But there isn't a lot on the concept of accelerated aging in vascular disease in addiction. So if you think about the aggregate of all the substances, the chronic use, the exposure, lack of sleep, nutrition, all of these factors, they feed into older adulthood, more so in adolescent and adulthood. In other words, there might be sort of an accelerated aging in addiction, and I'll be talking about that as well. So we care very much about deep phenotyping in the samples that we study, cocaine and heroin addiction. And maybe because we're psychologists, Rita and I, and so the way that we manage the phenotype is that we're not just interested in the presence or absence of the disease of addiction, but we're interested in variables that contribute to the severity of addiction, that contribute to individual differences in addiction. And in order to be sensitive to that as a scientist, you know, when you're working in your lab at Mount Sinai, in order to be sensitive to that, you need to be able to go to rehabilitation centers in Mount Sinai or outside and really be with these individuals, really see what are the different factors that contribute to their suffering. And so one of the organizations that we develop relationships with is Samaritan Daytop Village. They have over 40 drug and alcohol rehabilitation programs all over New York. But in addition to that, so in addition to providing, you know, addiction and mental health services to over 10,000 people every day, and of course at Mount Sinai, even more thousands of people every day are being serviced. But at Samaritan Daytop Village, they also house about 7,000 individuals every day. 3,000 of them are women and children. And they eventually find safe and permanent housing for people with addiction. And they found about 1,000 just in the last year. And so the organization is thinking about the 360 around the individual, not just the addiction, but also the other issues that plague people with addiction and people with other psychiatric disorders as well. And so I became a member on the board of directors of Samaritan Daytop Village. And over the years, I became the vice chair on their board of directors. And so I was able to steer the organization towards understanding the importance of research as something that will come back to their clients in terms of information and enrich their lives. And so we were able to establish within our data pipeline that you can see here, we were able to establish a situation where we have lab space within Samaritan Daytop Village rehabilitation facilities, and we can start collecting some of the data. And we can also have our postdocs and other students in our lab getting the exposure to the phenotype that we're studying. And that is very important in terms of generating ideas, generating questions, and also generally developing your sensitivity to the suffering of other people who are of much lower socioeconomic status than you are. And these are the samples that we're studying in addiction. Generally, we study people with very low socioeconomic status, people who suffer from a lot of other things in addition to addiction. And so then we can, after we have deep phenotyping, we can actually refer them to all of our specialized and sensitive and complex imaging processes. And so what I'm going to show you, results I'm going to show you, have to do with a structural MRI scan, voxel-based morphometry, functional MRI, the Bolt signal, but also PET studies where you can attach a ligand, you can develop a ligand that can map certain chemicals in the brain that sometimes cannot be done using MRI. So I will start with women. Women are very important, obviously, very important part of the population. It's not just that they're 50% of the population, but they are a population in itself. And I'd like to make just 2 points before I start with data. I'd like to say that women are not reducible to menstrual cycle. So for example, this is a figure I lifted out from a review paper by Yevin Shaham's group, where they are reviewing in human and rodents the effects of sex differences, different sex difference studies, where they highlight the high cocaine craving during follicular phase in women versus the luteal phase and in rodents as well. But women, I don't know about rodents, but women are not reducible to their menstrual cycle. And there are a lot of other issues, including biological factors that happen before one gets their menstrual cycle that can affect the addiction, the onset of addiction, but also the severity of addiction. And there aren't too many studies. Women are also not reducible to sex differences. And so I'd like to bring your attention here to this lower panel where you can see together with Anna Zilverstan, we reviewed pretty much all of the fMRI studies that were done at the time in addiction. And you can see there were altogether about 100 studies, a little bit more than that. And you can see here that, for example, about 32% of these studies did not include females at all, and about 42% included less than 10 females, so not allowing for sex differences to be studied at all. But pay attention to this really small red sliver here that shows that only about 2% of all the studies we reviewed had 100% females. And that's what I mean when I say that women are not reducible to sex differences. Women are a population in itself. And so they need to be studied at least just as well as much as men in a focused way as a population in itself. And that is a very important point that I always bring up when people argue with me that there are no sex differences. Well, there are sex differences, and sometimes there are no sex differences, but that doesn't matter. It matters to study women. And so one really good target, a really good marker, if you will, of sex and sex differences is aromatase. Aromatase is an enzyme that irreversibly converts androgens to estrogens. And so here, if you're interested in the chemical process, on the top panel here, you see that just a general reaction of the conversion by aromatase, of the conversion of testosterone to estradiol, and and that's done by the catalyzation or aromatization. And that's why I guess it's called aromatase. In the second panel here, you can see the catalytic mechanism of aromatase, which involves a methyl group that is oxidized and subsequently irreversibly eliminated. The inhibition of aromatase lowers the absolute levels of estrogen and may increase testosterone. And so reducing estrogen-testosterone ratio both in men and in women. And this ratio, as we know, is very different between men and women. Aromatase inhibitors pharmacologically can be, for example, Vorazole, are used for many different indications, including breast cancer, but not just. But there are also drugs that inhibit aromatase, as if it was a pharmacological dose. And so smoking is one, for example, one drug that is used before the onset of the main addiction that we study, that chronically reduces aromatase availability in the brain. So all of these things are likely to have sexually dimorphic function and functional outcomes. And so it's important to remember that. When I was at Brookhaven National Laboratory, I was able to witness the development of aromatase as it's being tagged and delivered to the brain so that we can map aromatase availability in the living human brain by tagging it with the aromatase inhibitor Vorazole. Now, we're not giving them the aromatase inhibitor Vorazole. This is just tracer level so that we can map the distribution of aromatase in the brain. And so you can see here in the brain images here The distribution of aromatase in the warmer colors, so the yellows and the reds, and everything that's in between. And here, this is at the level of the thalamus, and this is the level of the bilateral amygdala. And you can see here that there is a lot of uptake in the thalamus, in the amygdala, also in other brain regions, but this is where the uptake is most visible for this particular PET tracer, but also other studies show that this is really the akin to the distribution in the brains of humans. I was able to go in to this study and just deliver my cognitive tests. And so what I chose to deliver was to just test whether a test of cognitive ability that is estimated as intelligence, how is that related to aromatase availability in the brain, and also memory, and also the multidimensional personality questionnaire, where specifically I wanted to know about constraint, so the ability to self-control. And these tests are based on other studies in the past in animals, preclinical studies that show a relationship between aromatase availability and cognitive ability, memory, including memory, but also personality measures such as restraint, or in this case, in humans, it's called constraint. And so what we found was that aromatase availability in the amygdala was related to cognitive performance, but it was related to cognitive performance only in men and not in women. So you can see here on the left, the list of tests, matrix reasoning, and rat word reading. These are tests to estimate intelligence. Also the CVLT, the California Verbal Learning Test, where you have to recall, after a short delay and a long delay, you have to recall the list of words that were read to you. And so you can see here the pretty serious negative correlations. And you can see that there's nothing here in the women. I'll show you the scatter plots. So in the scatter plots, you can actually see that men with higher performance of memory, so their ability to retain many more words than others, had less aromatase in their amygdala. And also men were able to do really well on the matrix reasoning, have really high estimated intelligence, or relatively high estimated intelligence, also had less aromatase activity in their amygdala. And so what does it mean to have less aromatase activity in the amygdala? We don't know that yet. It might mean having more or less testosterone, or having a change to the testosterone-estrogen ratio in these men. Also, these are non-smoking individuals. They don't suffer from addiction. And so here we wanted just to know the baseline activity of this ligand in the brain. So aromatase availability in the amygdala is also associated with trait constraint. So the degree in which you're able to self-regulate and self-control. And in this study that I'm showing you, that was published not long ago, these are individuals with obesity. And so it shows here that the more constraint that they show, the more aromatase availability in the amygdala. So how that is connected with their cognitive function is not clear yet. These are tidbits of experiments that have to be continued in order to flesh out the causative variables. So what's important also to note, and I've noted it before, is that aromatase is being inhibited, especially by chronic smoking. So smoking of one cigarette is not enough. But when there's chronic smoking, there's inhibition of aromatase to about 30% in humans. This is an early study that Anat Biegon and her group did in baboon, in a female baboon. And you can you can see here the MRI scan, you can see the baseline PET scan. So you can see the distribution of aromatase here in the bilateral amygdala, very similar to humans. And you see the inhibition with nicotine. And that is true for humans as well. Aromatase is also significantly inhibited by methadone. Methadone is a potent inhibitor of aromatase, and it alters endogenous testosterone. So, for example, there's low bone density, and that may be due to low estrogen concentration. There's low bone density in about 83% of patients on the methadone maintenance treatment program. And also the side effects of methadone, which include flushing, muscle pain, can be in part explained by the drug's inhibition on aromatase. And other drugs also, alcohol, also have effects on aromatase. So all of these things make us ask ourselves, what is happening also in terms of sex and in terms of gender to people who are on a regular basis, their aromatase is inhibited. I'm going to shift now to talk to you about gray matter, structural changes as a function of sex. This is a study that we conducted with Rachel Rabin, who was a postdoc at the time. Now she's in McGill. So this is a voxel-based morphometry study from the Enigma Addiction Consortium, where we took T1 weighted MRI scans pulled from 7 sites. So we had 420 participants, people with cocaine-dependent men, cocaine-dependent women, and control men, control women, very nicely matched. And what we see for women, involvement in the insula, in the anterior insula. So such that women with addiction had less gray matter in the anterior insula than control women. So that's an effect that's specific to women in gray matter. And it makes you think about interoception, about cravings, because the anterior insula is associated with these processes. But what affects men is reduced gray matter in the hippocampus, and that is directly affected by duration of lifetime cocaine use. So men with less hippocampal gray matter also had more time using cocaine. So more years of cocaine use, less gray matter in the hippocampus in men, and there doesn't appear to be the same effect in women. We also conducted fMRI studies using monitor reward. And here we mapped diagnosis, sex main effect, diagnosis by sex overlap, and diagnosis by sex interaction, while men and women were doing a task, actually a Stroop task with monetary conditions. So if you look at I'm going to just focus on the diagnosis by sex interaction. And you look here in the inferior frontal gyrus, here in yellow, you can see that women have more response in the inferior frontal gyrus to the higher monetary reward. Money equal the ability to procure drug in this case, and this is how we interpret it. Because we were also able to see that from baseline to after the task, the self-reported craving delta, so the change in craving following the task, was correlated with this particular inferior frontal gyrus response to 50 cents more than 0 cents. And it appears to be especially apparent in women with cocaine use. So the more craving they report, the more was their response in the inferior frontal gyrus. Other studies that we've had even before that in women with cocaine addiction, you can see the small sample size. We had them watch a movie about cocaine use, and another movie that was just neutral, was a nature movie. It was just as interesting, but it did not involve people using cocaine. And so when you compare men to women, you can see, I mean, the interpretation, the way I'm going to read it to you, is that women significantly decreased metabolism in the executive networks by up to about 8% when they were watching the drug cue movies, whereas men tended to increase metabolism to the cocaine cue movies versus the neutral movies. So again, this is interpreted as a limited ability to restrain and to use the executive functions. Another variable that's very important that also can be premorbid, if you will, is the impact of childhood trauma, which is a very terrible source of stress at childhood. So the impact of childhood trauma has been studied, you know, in individuals, not just with addiction, but also with other disorders. And it's been mapped in terms of brain regions that lack gray matter, for example, as a function of childhood maltreatment. And so we wanted to see what is happening in our own sample. And so this is work we did with Karen Baki, who was a postdoc at the time with us. Now she's faculty here at Sinai. And so we parsed our groups to people with cocaine addiction and low trauma, cocaine addiction and high trauma, versus controls with low trauma. And so we had these three groups, and here already you can see that a little bit less education in people with high trauma, which may have to do with the time of the trauma being premorbid, interfering with education. But also they seem to report less constraint, less ability to self-regulate, and more symptoms of current depression symptoms. So irritability, inability to sleep, and things like that that you can see on the Beck Depression Inventory when we asked them what happened in the last 2 weeks. And so what we find is that cocaine addicted individuals with high childhood trauma have less gray matter in the orbitofrontal cortex, in this region of the ventromedial prefrontal cortex and ventrolateral prefrontal cortex. And you can see almost a dose effect here. Here is the scatter that really nicely illustrates how there's almost a dose effect where you have the controls here, we have cocaine addicted individuals with low trauma and cocaine addicted individuals with high childhood trauma, having the least gray matter in their prefrontal cortex. But that's not the whole story. The same regions in the prefrontal cortex are also sensitive to the monoamine oxidase genotype. So again, genetic polymorphisms are very important to study in addiction and in other psychiatric disorders because they parse the population in a way that you can check to see if there are relationships there to the disease. They're not as rare as deletions, for example. And so you can really see what's going on as a function of the genotype. And the monoamine oxidase genotype is important in the sense that it's been linked before to externalizing psychopathology, including anger and aggression, but also to the risk for developing addiction. And so we decided to test whether the low versus the high allele of the MAOA gene was related to gray matter in our population of people with cocaine addiction. And indeed, we find that people with the low allele, which is the risk allele, the low MAOA genotype, have less gray matter in the orbitofrontal cortex, very similar to regions to where we found the effect of childhood trauma. And we find that people with a high MAOA genotype have very similar gray matter concentration as controls. And so what we call it is gene by disease interaction. Because here, yes, we can say the MAOA genotype, the genotype itself a person is born with, but there's a continuous effect of the genotype, including when on the onset of addiction perhaps, but also after the individual is addicted already. But that's not the whole story, because what we find that on the same region converging here, the orbitofrontal cortex, there are other effects as well. There's effects of cocaine use. So there's an effect of lifetime cocaine use and an effect of the MAOA genotype. And all the other brain regions, you know, this is a table of the contribution, of the relative contribution using multiple regression. The relative contribution of these brain regions and lifetime cocaine use, lifetime alcohol use, lifetime smoking, and the genotype. So you can see here that for the orbitofrontal cortex, the more years of cocaine use, the less gray matter these individuals had. And also the more years of cocaine use, the more years of smoking, the more years of alcohol converge in this particular region, the hippocampus, in the sample. So it's important to parse out these individual differences. But it's also important for me to tell you that although I've showed you decrements in the orbitofrontal cortex having to do with variables that might be pre-morbid, in this comparison and in many other comparisons we've done before and we will do after, we find that the prefrontal cortex is gaining while people are going through rehabilitation and while people are abstaining from drug use. So this is VBM analysis that we did with Muhammad Parvaz, was at the time a postdoc with us, and now he's faculty here at Sinai. So what he did here is do VBM analysis at baseline at 6 months follow-up of individuals who went through rehabilitation and abstinence. And so you can see the gain in the prefrontal cortex, the orbitofrontal cortex, or the ventromedial prefrontal cortex, and the inferior frontal cortex, inferior frontal gyrus. So you can see that after 6 months, the same individuals had more gray matter in these regions. But what does it mean to have more gray matter in these regions? I mean, it's not enough to show this gain in terms of gray matter. Very important for us to go back to behavior and see if behavior has fared in those 6 months, and indeed it did. So here is a good example. This is a task of decision-making where people learn implicitly that there are disadvantageous decks and there are advantageous decks where if you pull cards from them, you get, at the end of the day, you get more money and you gamble less. It's called the Iowa gambling task. And here you can see that for cocaine-addicted individuals, and not for controls, but for cocaine-addicted individuals, 6 months later, they not just had more gray matter in their orbitofrontal cortex, but they were able to make better decisions. And so we see the heterogeneity of the orbitofrontal cortex in human addiction. There are all these, this is just the blobs that we found over the years and over many different studies that involve, in addiction, that involve the MAOA polymorphism, childhood trauma, treatment with methylphenidate, response to money, extinction of drug use, insight and awareness, biased drug choice, and gray matter recovery, as we showed you. So it's very important that a lot of the heterogeneity, a lot of the individual differences converge on cortical regions, cortical prefrontal regions. And it brings again the emphasis and the importance of studying humans. Okay, so age. Age is another variable, and we don't pay much attention to it, even in animal studies of addiction. Very often, you want to make an experiment to see an effect of a drug on some other measure, recovery or drug use. Then you will use mice or rats that are, basic adults, young adults. In our populations, we use the people who come to us to do studies. The people who are already in rehabilitation, some of them are in the streets, but some of them are in rehabilitation. They are in their 30s and 40s. And so what we get is adults, and then all the other age groups are being ignored by us and by other labs. And so one concept that I've been studying that we reviewed extensively with Karen Baki was accelerated aging in addiction. And so first of all, we wanted to learn what is accelerated aging. It turns out that it's been talked about, and there are many studies generally on accelerated aging. And there are ways to compute accelerated aging. And so it's important to bring that literature and to bring that knowledge into addiction. And so let's look at the progression here of the some of the variables that contribute to accelerated aging and that are especially important perhaps in addiction. So you have the genome, the environment, and development, addiction to illicit drug use, tobacco, alcohol use, but also poor nutrition, poor sleep, no exercise, poor treatment, low socioeconomic and social psychological factors. All of these contribute to accelerated aging. And so why? Because they contribute to oxidative stress, molecular and biological and epigenetic effects. They contribute to brain disease and lower cognition. They contribute to certain behaviors. And they contribute to inflammation in the brain and in the periphery and to other comorbid disease. So there's brain decline in volume and function. There's early onset of all kinds of diseases like cardiovascular disease and also other organs. I just want to check if I'm good with time. Yes, okay. And all of these lead to premature death, but also to just aging that is accelerated. So we decided to focus on the vessel, the effect of cocaine on the vasculature. And so if you map, and Karen Baki did a really great job mapping the effects, the acute and the chronic effects of cocaine on the vasculature. And we're most interested in the chronic effects, of course, because our subject had more than 15 to 20 years of cocaine use and other drug use, and that in turn affects their vessels. And so we decided to focus on the carotid arteries. The internal carotid arteries relay blood to the brain, especially the prefrontal cortex of the brain. Okay. And so if you look at what happens if you look closely at what happens inside the carotid artery, you can see an example here that I put of a plaque burden. And so the carotid artery changes shape as a result of aggregated inflammation that becomes plaque and over time occludes the internal carotid artery. But even without occlusion, even if you think about people with cocaine addiction who do not have occlusion of the artery, in other words, they're asymptomatic for any cardiovascular or cerebrovascular disease, maybe, perhaps, they have aggregated inflammation, and that in turn affects brain function and behavior. And so we set out to study that. And we were able to create collaborations with Zai Fayed from BIMI, and he is one of the people who developed and studied very closely the inflammation in the arteries, not just the carotid arteries. And so with PET, with FDG, you can actually study not just glucose metabolism in the brain, but also the presence of inflammation in the carotid artery. You can also image the artery, okay, with MRI to study the thickness of the artery. And at the same time, if you have a PET MR like we do at Mount Sinai, you can also image the brain. So you can get a very rich description of neck to brain effects. And in this case, in the carotid arteries, there's reason to believe that these are effects that are important to brain function and behavior. And so what we did is we used, there's a group of people with cardiovascular risk. Their mean age is about 64 years old. So this is a sort of a convenient sample that we already had from Zai Fayed's Laplace study that was published in Lancet. And then we collected more individuals, people with cocaine use disorders, and also healthy controls. And you can see the age difference. People with cocaine users are in their 50s. You can see also the difference in lifetime nicotine use. So the convenience sample of cardiovascular risk, they're non-smoker and they don't have addiction. Healthy controls do not have cardiovascular disease nor addiction. And then cocaine users who are asymptomatic, considered to be asymptomatic to cardiovascular disease, but that's because they were never really studied or tested. And what do we find? Well, we find that people with addiction have more inflammation in the carotid arteries than people with cardiovascular risk. So here these are two individuals, one with cardiovascular risk and one with cocaine addiction, and you can see it, it's visual, you can see the difference. But also when we scan the whole group, the whole sample of individuals with addiction, we find that most of them have inflammation at the clinical level, inflammation that deserves clinical care, clinical attention. We also find that people with cocaine addiction have more plaque in their carotid arteries, and this is done with 3D dark blood MRI of the wall area and the wall thickness. So they have a larger wall area and more thickness in their carotid artery, indicating the burden of plaque. And what was interesting for us to find was that burden, so the more burden of plaque in the carotid artery, the more people reported cocaine craving and cocaine severity of withdrawal. So you can see that in that scatter. But we also find that correlations of this plaque burden with alcohol lifetime use and with systolic blood pressure. And these are individuals with addiction who are asymptomatic for cardiovascular or cerebrovascular disease, which means they will never see a neurologist. And then, of course, to us, what's interesting is how the inflammation in the carotid might affect the bold signal, the brain signals that we often look at. And so we entered the inflammation markers of the MRI and PET as a seed in connectivity analysis, resting connectivity in our sample of cocaine-addicted individuals. And then we find that inflammation in the carotid is connected to increased connectivity and decreased connectivity. So that the more inflammation in the carotid, the less resting connectivity of the dorsal anterior cingulate, the inferior frontal gyrus, and the more connectivity in the caudate and other striatal regions. And here you can see the scatter plot showing the resting connectivity in the cingulate area and wall area in the right carotid. And you can see this, the scatter, and it's probably, it looks very nice, probably because it's a very small sample. So I'm going to summarize by telling you, asking you, what are the factors that contribute to addiction severity? Pretty much all of them. And it's now with big data, with our ability to use computers and deep learning and all of these wonderful, wonderful methodologies. Now is the time to look at individual differences. And if you were sensitive to individual as a PI, if you were sensitive to individual differences from the get-go, then right now you probably have a huge data set of people, with a lot of individual differences and I know that in addiction, we do have this case. And I want to bring your attention also to a paper by Anna Silverstand, where she does this study in cannabis use disorder. I'm very proud of her for that. I also want to show you, in terms of acknowledgement, some of the people who helped a lot in making these studies possible. So the Brookhaven National Laboratory, these are people who developed the PET scans, including FDG and the aromatase and the MAOA. And I learned a lot from them, still am learning from them. And also our group in the picture. Unfortunately, we don't have the current group in the picture. I have the previous people, because they really contributed to this data. And so you have Karen Baki, Anna Silberstein, you have Muhammad Parvaz. Of course, Rita Goldstein doesn't need any introduction. So these are all the researchers that contributed. And from our funding portfolio, you can see that we're still very much interested in sex differences in cocaine addiction. So we're going to continue with that, and with the atherosclerosis in cocaine addiction. So that's a recent grant that we received. And I think that's it. I hope I left some time for questions. And so thank you for your time.
Thank you very much, Nelly. That was really, really interesting and very impressive cohort of work. And we have like 3 minutes for questions. So anyone who wants to throw in something, please put it in the Q&A box. As the host, I actually thank you for speaking the whole through almost through the hour, because then my hosting roles have shrunk a little bit. I'll start with one question that you mentioned briefly at the beginning, which is the role of, and I like it that you keep this, the abuse, neglect, stress as potential risk factors or antecedents of addiction. And you mentioned that neglect is not studied. Are you doing something about it, or thoughts of why not?
Perhaps it's more difficult to study, perhaps it's more difficult to define,
right.
Or perhaps people just don't think that that it's important enough. I don't know, but I think neglect is a huge important factor for many different reasons, including the issue of isolation,
right,
and social isolation. That has a lot to do with neglect. I think I saw Dolores Malaspina have her hand up?
Let me see. There,
Did I
no,
see that?
there is a Q. There is one question that was dropped in, which is about distinguishing pro-inflammatory phenotype as a cause, or its relationship to substance use and abuse. Is that
Hasn't
something?
been resolved.
Yeah.
That's why I studied.
What's the thought about in cytokines in
Yeah, I mean,
this developmental course kind of map that you're showing? How would you think
well,
about it? Where would you put it in this?
everywhere. I would put it everywhere. And why? Because it has to do with increased stress. It has to do with poor sleep. It has to do with trauma, childhood trauma. All of this, what I'm saying, it has to do with, there are papers behind it. There's data behind it, preclinical and clinical data. So it's not just the addiction by itself, which it does affect inflammation, cocaine and heroin and alcohol, but it's not just the addiction itself, but it's also addiction phenomenology. So,
and one last thing, because the beep here says we are almost on the hour. The series of studies on aromatine and the sex differences in those findings, the results were very strong and impressive. Any thoughts of using those kind of molecules for treatment in men?
yeah,
Or targeting that particular pathway?
Well, using aromatase inhibitors in men is undesirable because it's feminizing.
Oh, well.
No, but
Okay.
it's true. It's not. It doesn't feel good. You know, it
Right.
causes hot flashes. It causes their voice to change. It really scares them.
Okay.
You know, to be a woman, to take something that
To be a woman scares men.
to become
Okay, that's
like
the
a
conclusion
woman,
of the
that's
talk.
not, doesn't sound good, right?
Okay. All right. So we are on the hour. So thank you very much. It was fascinating. And please keep on this kind of work. And we'll see you again in a couple of years with some more exciting findings, I hope.
Sure. Thank you very much.
Thank you, everyone. Goodbye. Have a great weekend when it comes.